Welcome back to Stem Cells Unveiled from Basics to Breakthrough. I'm Dr. Sukant Khurana, your host, and we are diving deep into episode 2, the biology deep dive how stem cells work. Returning with us is Basudha Banerjee, a stem cell regenerative medicine biologist, a very young scientist who brings a lot of enthusiasm, a lot of modern knowledge to the table. Basudha, take it away. I'm excited to have you here once again, and I'm excited to unpack this.
Many thanks, Dr. Khurana. I'm so thrilled to be back. So, last time we laid the groundwork on what stem cells are, and now I want to get under the hood. So, how do these incredible cells actually work? I mean, that how do they divide, differentiate, and keep all our tissues ticking? And that is exactly what fires me up. So, let's start with the cellular mechanisms. And to me, it is all about their life cycle. So, stem cells have this amazing ability to balance between self renewal and differentiation. And these two are the two remarkable properties that stem cells own. So, Dr. Khurana, how would you kick off that idea?
Glad that you're steering this ship, Vasudha. I would say it's their dual nature. Stem cells divide asymmetrically, often producing one copy to maintain the pool and one that differentiates into specialized cells. It's a tightrope walk of fate decisions.
Absolutely. And that asymmetry is the key. So, picture a stem cell in the bone marrow, say a hematopoietic stem cell that is generally found. So, it divides, and one daughter cell stays a stem cell ready for the next round, while the other one that starts down the path to become a red blood cell or a macrophage or a variety of blood cell. And that is self renewal and differentiation both simultaneously working in action. And the most important part is that it is not random. Signals like cytokines or growth factors, they nudge them along. I have always found it wild how they maintain tissues this way. Take skin, for instance. Skin have basal stem cells. They churn out new keratinocytes daily to replace what we shed. It's a constant renewal engine.
Yeah, I love that renewal engine. It's like they are unsung heroes of homeostasis, keeping tissue stable over decades.
Totally. And this brings me to the machinery inside them. So let us zoom into the organelles and structures that make this possible. The nucleus we previously discussed is the command center, housing the DNA that dictates whether a stem cell stays pluripotent or whether it commits to a lineage. I mean, the transcription factors like NANOG or OCT4, they are sitting there ready to flip switches. So, what is your take on the nucleus role, Dr. Kurana?
It's the master regulator, no doubt. Chromatin structure in the nucleus, how tightly or loosely DNA is packed, controls gene access. In stem cells, it's more open, flexible, which ties into their potential.
That is right. So that euchromatin state is what keeps them poised for action. And speaking of action, let's not sleep on mitochondria. I'm sure it's your favorite. So in stem cells, they're very fascinating. They tend to rely on glycolysis over oxidative phosphorylation, even when oxygen is around. It's like they're in a low energy protective mode to sort of avoid the DNA damage caused from oxidative stress and all the ROS generation. And I've also seen few studies where mitochondrial activity it ramps up as they differentiate, say into neurons or muscle cells. So did you ever dig into that metabolic shift?
Yeah, I am very interested in metabolic shift. Mostly, it's in two different contexts. We have spoken about things in context of cancer, Warburg effect, right? But I'm very interested to dig that into even the context of pluripotent stem cells. Also thought of some metabolic shifts in the context of ER structure and how complex they are. But this is very interesting. Metabolic flexibility is the clutch. It sort of preserves the stemness until they're ready to specialize. Then mitochondria kick into high gear to meet energy demands of differentiated cells.
Exactly. And it seems such a cool transition. And while we are on organelles, the cytoplasm worth a nod. It's where all the signaling pathways play out, integrating the external cues that tells what the nucleus has to do. Then we also have the cytoskeleton, microtubules and actin filaments, which sort of give stem cells the shape, mobility, especially when they migrate to repair tissues. It seems also interconnected, and it definitely is. But here's what I want to pivot. How do stem cells stack up against specialized cells? I see them as plastic, moldable, while a neuron or cardiomyocyte is locked in. So, what is your lens on that comparison?
You're framing it perfectly. Stem cells have that plasticity, open chromatin, metabolic adaptability, while specialized cells are terminally differentiated with fixed gene expression and structure. A neuron's got dendrites, a cardiomyocyte is packed with myofibrils. Stem cells don't commit until signaled.
Yes, that is it. So, plasticity is their superpower. Pluripotent stem cells, say, can become anything, liver, lung, you name whatever, because its fate isn't sealed at the moment. Compare that to a red blood cell, which ditches into the nucleus, which basically ditches its own nucleus entirely to focus on oxygen transport. So I sort of love how stem cells hold this potential. It's like a blank canvas, while specialized cells are the finished painting. And most importantly, it is not just about the embryonic stem cells. The adult ones, like the intestinal crypt stem cells, they still have enough plasticity to replenish specific tissues over a lifetime. It is mind blowing how they toggle between these states.
Mind blowing indeed, that toggle maintaining plasticity yet responding to cues. It's what makes them so versatile biologically and therapeutically.
Therapeutically, yes. Now we come to the translational thing. So that's the hook for me. Understanding how these mechanisms open doors for harnessing them further for translational and clinical applications. So imagine tweaking those signals, or rather organelles, to push stem cells where we want them, like repairing a damaged heart or brain. So, do you have any final thoughts before we wrap this deep dive?
Yeah, just that you painted a vivid picture, Vasudha. The biology is the backbone for everything we'll explore. Applications, breakthroughs, all stemming from how these cells tick.
Glad you think so. Honestly, I could talk stem cells guts all day. But let's save some time for the next time. And thank you for letting me lead this, Dr. Khurana. It's been a blast geeking out with you.
It's my pleasure. That's episode 2, folks, led by brilliant Basudha Banerjee. Next up, we'll explore where stem cells come from, sources, and harvesting. If you have got questions, you've got comments, suggestions, please type them in the comments section. Please subscribe to us. Let us know how we are doing. Please send us suggestions. We'll incorporate them. Until then, thank you. It was nice having you, and thank you, Basudha, for being here.
Thank you. Thank you very much.